Okay, so guys, basically, when I talk about biotechnology, there are going to be two terms which are going to be involved out over here. First, that is bio, and second is technology. Okay, bio means what? Okay, basically, just to give you an idea or to speak in a layman's term, the screen is blurred. Okay, is the screen blur for everyone? Just give it a couple of minutes. I've just started the screen share process, right? Okay. Okay. So, guys, basically, when we talk about biotechnology, what do I really mean by biotechnology? See, bio in a layman's term, okay, in a completely layman's term. Okay, 1 minute. What I'll do is 1 minute. I'll just stop the screen share and I'll once again restart it. Okay, now I have once again restarted it. Okay, so now just see. Okay, chalo. So basically, in a layman's term, guys, what is biotechnology? Biotechnology is nothing but it is the use of biological organisms. Okay, use of biological organisms or use of your biological entities for creating substances which are going to help mankind or which is going to help in the betterment of humans. That is nothing but your biotechnology. So use of biological organisms for the betterment, okay, for the betterment of the system, for the betterment of your human race, for the betterment of mankind. That is nothing but your biotechnology. But now, guys, the question arises now. These days we have been talking about, oh my god, oh, biotechnology, this is a upcoming field, it's a booming field. So is it really like a booming field? Is it really like a new field, or has it been existing since ancient ages? Yes. um, Guys, the thing is, biotechnology is an age old industry. Okay, it is not something that is new, and we use these principles of biotechnology in our day-to-day lives, guys. Okay, for example, even when we look at preparation of curd, okay, you guys must have made curd at your place. So what do? See, first thing that you're going to take is you are going to take a spoonful of already prepared curd, right? And you take that and you are going to put it into a bowl of milk, right? And then you're going to just mix it, swirl it, and then you're going to just keep it aside. What happens immediately? It is going to convert into your curd the next day. So what is this, guys? You are employing bacteria to give rise to a biological product. What is that? That is nothing but biotechnology. Okay, let's say we are talking about a bread. Even for bread making, guys, you are going to require yeast, right? So you are employing biological organisms, biotechnology. Okay, for example, guys, wine, you are going to employ yeast. Okay, so once again, biotechnology. For cheese, once again, biotechnology. So guys, when we talk about biotechnology is an age old process. Okay, however, in recent times what we have done is we have added onto it and we have increased its ability to help mankind. That is what the change has been. Okay, so guys, once again, what is biotechnology? It is nothing but the use of biological organisms for the betterment of mankind, the use of biological organisms or their products. Okay, whatever that originates from biological entities, that can be used for your humans for their betterment. Okay, so let me just put down the definition out over here, guys. So okay, so we have different bodies out over there who give us different definitions. Okay, and few of these definitions, they are very very important. You have to keep them in mind. Okay, so The first definition basically is the development and utilization of biological forms, products, or processes for obtaining maximum benefits to humans or other forms of life. Okay, so this is the first thing. Okay, but now, guys, there is another entity, okay, whose name is OECD. Okay, so this is normal definition, but according to OECD, okay, and what is this OECD? Organization for Economic Cooperation and Development. Okay, so according to this OECD, guys, we have another definition out over there. So let's try to see what is this definition. Okay, so basically, according to OECD, uh, if I zoom in into this, the font will be extremely huge, guys. See, if you feel still it is okay, then I'm fine with this. This is what a webinar have they started? Like, is this a webinar kind of thing? Is this normal lecture or a webinar? Oh, okay. So it has that side by side view, right? Okay. Yeah. Okay. Anyways, let's try to see according to your OECD. What does OECD say? OECD is nothing but it is going to talk about like what is biotechnology? It is nothing but the application of scientific and engineering principles to the processing of materials by biological agents to provide goods and services to human welfare. Okay, so that is this thing. So basically, you are using the organisms, and along with organisms, you're going to be using their parts or products to help in this thing, to help your humankind. That is nothing but your biotechnology. Okay, so guys, these two definitions you need to remember because they will ask you, like, for 1 month, define in your board exams. Okay, guys, according to OECD, what is biotechnology, or what is in general defined biotechnology? That is it. Okay, but now, guys, when we talk about biotechnology, how are we going to really see, like, what is exactly happening in biotechnology? Is it a separate field altogether, or is it like an amalgamated field, or is it, like a standalone field? So, guys, when we talk about biotechnology, it is not a single field as such. Basically, it uses different fields or inputs from different fields. So it uses principles. Okay, so principles of many fields. Uh, okay, it makes use of principles of many fields. Like, for example, guys, we have microbiology. So it talks about microbiology, principles of it. It is governed by that thing. Then it is going to make use of biochemistry. Okay, microbiology, biochemistry, because see, we need to understand the processes, like what is exactly happening inside the organism, like microorganism or the fungus or the algae or the yeast. Okay, or your your bacteria, what exactly is happening in that. Guys, after that we need to make use of statistics. Okay, because we need to understand whether our results are proper or not. Does it make any sense? Is it like, important? Is it not important? Whether we are getting the right stuff, whether we are going in the right direction. Okay, so that way. Apart from that, we require computers. Why computers? Because all these statistical thing and your softwares and all, they are going to be run by computers. So you need to know, have a knowledge of this. Along with this, guys, for your fermentation and all, we require all the other industrial processes. So, again, these are a few fields that we have spoken of. Like, then it can be your food engineering, then it can be chemical engineering, it can be mathematics, it can be, other fields as well. All of them, okay, all of them basically are going to be used in order. in order to give rise to your biotechnology. Okay, fine. Okay, so basically this is the idea behind it. But now let's try to talk about, like, what exactly is biotechnology? How is it really going to be, like, what really happens out over there? Okay, so guys, when we talk about biotechnology, first we'll talk about traditional biotechnology, how it was in the earlier days. Okay, so basically, when we talk about biotechnology, guys, we have two types of biotechnology. One, which was traditional, age old biotechnology. And the second one, guys, this is going to be your modern. So, what are the recent changes? What has really happened? Okay, but now traditional biotechnology, guys, I just told you, you have been using it in your household things over and over again. You know, this is going to be useful in the preparation of curd. You have used it in the preparation of bread, cheese, okay, wine, etc. Okay, but now when we talk about one moment, huh? No, it cannot be possible because now the font size is so huge that I cannot adjust my writing and the other thing. Okay, so it cannot go on the same page. This font size is too huge. This is like 100%. I generally use around 80%. In 80%, it looks proper. Here, then I won't be able to write only if I keep the text for the top portion. Then see, if I keep the text, see, this is what will happen. I won't be able to write. I'll get no space at the bottom. Okay, but now in your modern biotechnology, what are we talking about, guys? We are talking about a recombinant DNA technology. Okay, now recombinant DNA technology. Yes, what is this, guys? In recombinant DNA technology, you are going to make changes in your genetic material, guys. Okay, so our idea is to change the genetics, add or introduce genes, our desired genes into microorganisms, and then let them grow that product for us. So, for example, guys, let's say you are not producing insulin. Okay, now see, earlier what had happened, for insulin we were dependent upon dogs. After dogs we were dependent upon cattle. After cattle we were dependent upon pigs. And then after, like, taking it from the pigs, guys, we were like, we need to find out another method where there will be no allergies, where there will be no cross contamination from other organisms. So what we did, guys, we moved on to E. coli. E. coli is your bacteria. What we did, we took insulin gene from humans, we put it into E. coli, and now suddenly we saw that E. coli started giving rise to insulin. We took that insulin, purified, and we started using it for us. Now, how did we achieve this? By recombinant DNA technology. Okay, so guys, basically when we talk about this thing, okay, so this recombinant DNA technology, always understand, this is the one which has the ability, okay, to change the genes. Okay, so this is going to recombinant DNA, it changes genes in an organism. Okay, one moment. Yes, yes, yes. Okay. Okay, so basically it changes the genes, or we can say, we are artificially modifying the genes. Okay, artificial modification of genes or modification of genome. Okay, now guys, I'm sure you must have come across the term genome. Genome is nothing but all the set of genes, everything, all the genetic material which is present in an entire organism. That is your genome. Okay, so through this recombinant DNA technology, guys, we are going to make sure that we can change the genes. Okay, now is there only this one recombinant DNA technology? No, guys, there are many other techniques out over here in modern this thing. Like, for example, the one that we are going to see, we are going to see polymerase chain reaction. Polymerase chain reaction is PCR, guys. Okay, one of the most powerful techniques ever. See, guys, these days, you must be seeing, oh, RT-PCR, RT-PCR, RT-PCR for COVID-19, RT-PCR, right? What is this, guys? Just to give you a layman's idea, let's say you have one fragment of DNA like this. Okay, one fragment of DNA, meaning a small gene. By the end of this polymerase chain reaction, guys, after 40 minutes, okay, in between 30 to 40 minutes, from one single copy I can get 1 billion copies, guys. 1 billion copies of DNA within just 30 to 40 minutes. This is like, a rapid amplification method. It's amazing kind of thing. That is your polymerase injection. We have to see this thing. Okay, guys, after this, we have something which is called as microarrays. Now, generally, microarrays, guys, you really don't have this thing, but they have just mentioned it. Microarrays is nothing but a chip method. Okay, a chip method to detect your DNA kind of thing. So you will have a chip, okay, on which you are going to have sample DNAs which are going to be present like this. Okay, there are sample DNA strands, single strands kind of thing. Okay, and now you take your sample, guys, and when you put it onto this, you are going to see that on this chip, in some cases, some DNA strands, they will be like pronounced more, so they will appear like this. Okay, now you don't have this technique at all, but I'm just giving you an idea. It's a very amazing technique. Okay, so when these particular strands of DNA, they are going to fluoresce. Okay, at that time we know, oh, these genes are going to be like working, meaning these genes are active more in your in a cell during maybe certain phases. So this is something called as microarray. Along with this, guys, in newer techniques, what have we come across? We take two different types of cells. Like for example, we take a potato, we take a tomato, we take this and yak kachak, we join them together. What do we get? Tomato, right? So what is that? That is cell fusion. So we are going to have cell culture, like, we are going to increase the cells, the number of cells, cell culture and cell fusion. So even that is there, etc, etc, etc. So guys, this is the differences. Okay, this is the difference between your traditional and modern technology. Yes, we get tomato. Yes. So we have Rafano brassica as well. So Raphanus and brassica, we both of them, take them together, we get your Rafano brassica that way. And guys, like that, we can bring about like fusion of or any kind of cells, any kind of cell. So this way, so basically, this is the idea behind, like, how this is going to be like, guys. Okay, but now if we are talking about, okay, biotechnology, like, like, who was the first person to really talk about, who are the people to like really work with this thing? Okay, so the first people to ever talk about this is your A. Kaiser Dell. Okay, now remember this thing. So this, when we talk about biotechnology, the first people to talk, okay, the first people to talk about it, okay, meaning to just give an idea, okay, about this thing. This is your A. Kaiser Dell and Peter Loban. Okay, so A. Kaiserdell and Peter Loban. These were the first people to ever work on this. Okay, and now the first people to prove, or to prove that, oh, that this can be done, whatever this Peter Loban and A. Kaiserdell think this can be done, this was Stanley and Cohen. Okay, so Stanley and Cohen. Okay, so guys, remember this name in your exams. These are always asked. So A. Kaiserdale and Peter Loban, and the second one is Stanley and Cohen. The first ones to prove. Okay, so guys, once again, like I said, when we talk about this modern day biotechnology, there are two aspects of it. Okay, So when we talk about this, first thing, it is uses different techniques, okay, which I have listed on the top. Okay, so biotechnology makes use of two things. Again, like different techniques. Like I said, we have PCR, we have microarrays. Okay, the chip method basically. We have recombinant DNA technology, which is called as rDNA technology. Okay, recombinant DNA technology is rDNA technology. So etc. Bioprocessing also we have, like we are fusing the cells together, and then we are trying to bring about fermentation, some kind of product formation. So this entire idea is to change the genetic material. Okay, so that way. But now, on the other hand, guys, see, one thing is you are going to use different techniques to change the DNA. But the second part, guys, is that you are going to have ownership of the technique. Okay, now ownership of the technique meaning what? You can patent this process. Like, okay, oh, you are having a new process, so you just own that, you patent it so that only you can use that thing. So that is called as ownership of technology. Okay, but now if you own some technology, like let's say you have patented some technology, it is going to come with some socio political impact. Okay, I will explain you what does this mean. Okay, so it comes with some social political impact. So guys, the first thing that I will tell you, okay, so when we talked about insulin, okay, now what happened was, guys, the people who were like producing this insulin, guys, okay, they could have very well patented it. Okay, and they would have said that, oh, that only we could use this thing, or we can use this. Okay, so now if they would have done that, guys, then what would have happened? Nobody else would have been able to use their product. Okay, meaning other companies could not produce it. Then what will happen, guys, is only one company will have the monopoly, others cannot produce it. So all the money will be going to only one company, so they can decide whatever price they want, and others will have to pay. Okay, so that is the thing of owning technology. Let me just give you another example. When I talked about this PCR, okay, polymerase chain reaction, this is like a wonderful just saw in GTB, they have given Stanley Cohen as one of the name. Isn't that correct? That is right. Uh-huh. No, 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 no. Okay, okay, okay. Uh-huh. Right, 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 right. No, no, no. It is right, right, right. Okay. So, sorry, sorry. No, no. It is Stanley Cohen and Herbert Boyer. Okay, no, this is not there for you actually. Okay, there were two people out over here. One is Stanley Cohen, Herbert Boyer. Sorry, I just made it like Stanley and Cohen and Herbert. Sorry, Stanley Cohen and Herbert Boyer. Okay, so now what happened with regards to your PCR, guys? I'll tell you. So there was this company called as Roche Chemicals. Okay, it's a very big company, very very big company. Okay, and mainly all the other companies used to go to Roche Chemicals to buy all the the ingredients, the reagents, okay, all that stuff. So now what happened was Roche Chemicals was working on this PCR, and suddenly Carrie Phillips, he found out that, oh, this method really works. Now what this thing did was Roche Chemicals tried to patent the technology. Patent the technology means what, guys? Nobody else would be able to use it. Or if you use it, so let's say, for example, like we have Shravani out over here. Shravani has given us some kind of a technique. If you want to use it, then you will have to pay Shravani some money. Okay, that is called as royalty. So basically, what this Roche Chemical did was it patented PCR. Okay, now everybody was like, this is such a breakthrough thing. It's a breakthrough in medical sciences. And if you patent this, nobody else will be able to use it. Then what is the use of this technology? Like, if you are going to mint it and you're not going to help mankind, will this be a real good thing? Roshi chemicals were like, nay, 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 I don't want to listen to you guys. I don't want to fall into your trap. I am going to patent this thing. So they patented this. But now what happened was others could not use the technology for the advancement of science. So now what everybody else decided was we will not order any chemicals from Roche Chemicals. Okay. Now, as the other industries, like all the other, people who were ordering chemicals and stuff from Roche Chemicals, they stopped their contracts. Now what? Suddenly Roche Chemicals was like, oh my god, it had not thought of it that this would happen with it. Now what? Now they said that okay, forget. Now we are losing a huge amounts of business, and that is going to go to some other companies. Ultimately, it is going to lead to a greater loss. So they said that okay, chalo, forget it. What we'll do is we will revoke the patent, and we'll allow everybody to use it in the benefit of mankind. So that is how it affects the socio-political impact as well. Okay, everybody all right with this thing, guys? So this is like see, it is always like that. With great powers comes great responsibilities. It's that way. So something of that sort. Okay, but now let's try to see this thing, like what is exactly going to happen in your modern day technology. Okay, so let's try to see what are the things that are going to happen. Okay, and the same thing, guys, with regards to your this thing also. Okay, so when we talk about this ownership of technology and all, so we have Alexander Fleming. Alexander Fleming, he found out penicillin. Now there was two options with him: either to use it, I mean patent it, or either not to patent it. But he decided, he will give it to the masses. Okay, so that way. So that is a very very noble thing, kind of thing. Okay, otherwise people can take in these days today, people take undue advantage of this patenting and all. Okay, so now modern day biotechnology, guys, it is going to be based on two main things. Okay, one is genetic engineering, where your entire aim is to change the DNA. Okay, it can be DNA, it can be RNA. Okay, and the second thing is chemical engineering. Okay, where you're going to look out for the fermentation techniques and all to increase the products. Okay, so now so now when I talk about genetic engineering, guys, once again, as I said, this is going to deal with all the alterations. You are fiddling with the DNA, basically DNA nucleic acid. So this comes with alteration of nucleic acids. Okay, now this can either be your DNA, most probably will go with this DNA. Sometimes we require RNA as well. Okay, so whenever we employ this thing, guys, what is this? This is called as your rDNA technology, or this is nothing but recombinant DNA technology. Okay, now this recombinant DNA technology, guys, this also in can be called as gene cloning. So where you're taking one gene and putting it into your another organism. So you're basically cloning the genes that you require. Okay, so if we talk about this, basically, these are the parts of it. But then let's try to see something more out over here, guys. Okay, so basically, guys, when we talk about genetic engineering as such, okay, so I will just give you the definition. So what is genetic engineering? It is nothing but the manipulation of genetic material, okay, so that we get a predetermined product. Okay, so let me just put this definition down so you will have an idea. Okay, so genetic engineering is defined as the manipulation of genetic material towards a desired end, that is your product, and in a directed and oh sorry predetermined way, meaning we are going to sorry okay. So, in basically predetermined way, meaning we have already found out, how we are going to bring about this process of like fermentation, or by making use of what techniques and all. So that is already been decided. So that is nothing but genetic engineering. Okay, but now who were the first person to like really work with this? Who were the people who actually talked about this? And how do we bring about this genetic engineering? What is the actual path? So just to give you a little idea. So guys, the first thing that it was done by John Smith. Okay, and what does this guy do? Now, John Smith was the person to, and this was like, basically, okay, he was nothing but the person who gave the definition of genetic engineering. Okay, so this was given by him in 1996. Okay, so what does he say, guys? Okay, now the definition is like, really, really huge. But just to give you an idea about what is he really talking about. So John Smith, what does he say is, when you are going to talk about genetic engineering, the best way to define genetic engineering is to transfer DNA. Okay, so he says you take the DNA, you put it into a vector. Now, what is a vector, guys? Okay, now for example, let me just tell you. A vector is someone who carries the germ, the DNA, or any substance from point A to point B. So let's say tomorrow you come down to Goregaon station and you take a rickshaw from Goregaon station to Wakade classes. Let's imagine. So guys, what is this rickshaw? The rickshaw is the vector. Okay, because it is not involved in any process. What is it involved in? It is involved in only the transportation of the DNA or the gene, but it is helping in transportation of you. So you take the DNA, you put it into the vector. Now this vector, what can it be? It can be your plasmid, it can be your phagmids, it can be cosmid, etc. Okay, we will see this thing. Okay, so for example, we are making use of plasmid. Plasmid is nothing but your circular DNA. So it comes like this. Okay, and in this you can add your entire gene. So let me just show you a quick idea. Okay, what is this? Okay, so let's say you have your DNA. Okay, so this DNA, this is your desired gene. Okay, so it is something like this. You take this gene and then you are going to insert it into this. So in the next case, guys, what are you going to see? You will see that this DNA, like the plasmid, it is like this. And now in this plasmid, now you have got your piece of DNA, meaning this can be your insulin gene, it can be any gene, whatever you wish. This is your DNA inserted into this. And now, once this is inserted into this, guys, after inserting this into your vector plasmid, you take this plasmid. Now, this DNA, whatever we have produced, this is called as your rDNA, that is recombinant DNA. Okay. Okay, now once you have done this process, guys, now you're going to take this plasmid and now you're going to put it into your host cell. Now, basically, host cell can be whom? Host cell can be your E. coli. Okay, because then why are you going to select like your E. coli or any bacteria? Because they grow at an alarming rate. Okay, it is exponential growth. One single bacteria can give rise to millions and millions of bacteria in one day. Okay, so it is that way, guys. Okay, and that is why you will always go with bacteria or this microorganism. They have rapid rate of multiplication. Okay, so now what happens? Let's try to see. So let's imagine this is your E. coli. Okay, E. coli has its own DNA like this. Now what are we going to do? We are going to take this, okay, and now we are going to put it out over here into this E. coli. Uh-oh. Okay, so we have taken this DNA, that is the plasmid, and we have put it into the E. coli. So guys, see, E. coli has got this thing. Now what happens, guys? This E. coli, now earlier, let's say we are talking about this gene as insulin gene. Earlier, was E. coli producing insulin? No, it was not. But now what happens is, guys, this E. coli, it starts producing your product, the desired product. Okay, it starts producing the desired product. And then this product, you can collect it from the medium, and then you can purify, you can filter, purify, then you can just market it, bottle it, market it, and distribute it. That's it. This is how your entire cloning and genetic engineering is going to work. Okay, everybody has got the idea how, or what is happening out over here. Yes. Okay, guys, in your textbook, if you look at this, this has been given in the worst possible manner. Okay, and that is why I want you to pay full attention out over here. What is vector in that? Vector is the plasmid. See this gola I have drawn. See this. See this thing. This is a plasmid. Okay, where do we get this plasmid from? We get it from bacteria only. So we have bacteria who produce this plasmid. Okay, and how do we get it? We just have to look out for those bacteria, and from those bacteria we remove the plasmid so that we can use it for ourselves. Okay, so just imagine, if you're thinking of it in that way, so just think कि ये rickshaw है, ये rickshaw, it is going to take your DNA and it is going to put it out over here. So it is just the transporting material. Okay, everybody all right with this? Fine, let's move ahead, guys. Okay, now see, this is what we need to know about your genetic engineering. But we said, the other part of this is chemical engineering. So, what is this really going to deal with? See, whenever we do all this thing, okay, so we said, oh, you take this DNA, you put it into the vector, you put it into cell. Can you do this outside on your, tabletop, or your dining table, or your kitchen counter? Can you do it out over there? Absolutely no. Nothing is going to work. You're going to get so much of contamination that your experiment is going to be 100% failure. So what we need to do in chemical engineering, guys, this is the method by which it is going to make sure that whatever environment that you're working is sterile. Sterile means what? It is free of microorganism, so that when you work with your microorganism, it is the only thing that is present. So generally, this is going to make sure of maintaining sterile environment. Okay, and this is done to avoid contamination, to avoid contamination from unwanted manner. Okay, so that is the idea. And now, guys, by doing this thing, what are we going to actually do? We are going to help in the production. Okay, so basically, chemical engineering, it is going to determine what are the parameters, what are the conditions that are required for the production of enzymes, or it can be your vitamins, or it can be organic acids. Okay, whatever we had seen in microbes in human welfare, guys, it's the same thing. Antibiotics, or it can be vaccines, etc., etc., etc. So this is what we need to know with regards to the difference between genetic engineering and your chemical engineering. Okay, so guys, just have a look, and then we move ahead with the next part. That is, if we're talking about this genetic engineering or this gene cloning technique or rDNA technique, guys, what are the different things that we are going to use and how are we going to achieve this? And then slowly we have to start talking about all those things. Okay, chalo. So let's move ahead, guys. Okay, so now when we talk about your the entire like gene cloning or rDNA technology, guys, we are going to make use of different things. See, because we cannot just take one cell and just take DNA from somewhere and just put it out over there. It is not so easy. Okay, in just explaining it out over here like this, it's simple, but in reality, it's not so. So what are we going to require? Okay, so for this gene cloning or recombinant DNA technology, rDNA technology, we require three important things. Okay, I have just given you an overview, like how we are going to do it. Okay, so I've explained you how we are going to do this thing. So guys, first thing is we are going to require certain tools. Okay, tools for your like gene cloning, meaning it can be instruments, it can be your enzymes. Okay, I will tell you. First, it is going to be your tools. Second thing that we require cloning vectors. Okay, cloning vectors, what did I tell you? See, this rickshaw kind of thing. Okay, so which is going to help you transfer from one place to another. So for example, plasmid. Okay, plasmids, etc. There are many plasmid, fachmids, cosmids. Okay, we will talk about them as we move ahead. And finally, the third thing we require is, see, this E. coli cell, right? I told you that you take this DNA, you put it into the E. coli cell. Arey, but this E. coli cell should be ready to accept. Okay, kal tomorrow somebody just gives you 1 million rupees just like that. Okay, पहले यू विल थिंक ना कि अरे बाबा ये तो 2 नंबर का पैसा तो नहीं है, ये ड्रग्स का मनी नहीं है, ये कुछ दूसरा मनी नहीं है, unnecessary, it wouldn't land you in trouble, right? So you have to be ready, right? You have to be ready to accept it. So these cells which we make them ready to accept this plasmid, guys, we call them as competent cells. These are called as competent cells. So these cells, they are actually ready. We make them ready. Okay, they are ready to accept the modified rDNA. Okay, they are ready to accept the modified rDNA. Okay, so these are the three essential components. But now, when I said we have tools, what is exactly included in tools? So let's try to see. Okay, so now guys, the first thing that we are going to use is we require a lot of instruments. Okay, because everything is not going to just happen on its own. So we require certain instruments, like I said, we have PCR, we have gel electrophoresis, we have your like your DNA, like your Agarose Gel Electrophoresis, your page, that is polyacrylamide gel electrophoresis, your chromatographic techniques. So we'll talk about that. Okay, and the second thing that we require is your enzymes. The enzymes, we call them as your biological tools. Okay, and biological tools, guys, we are going to make use of enzymes. Okay, and enzymes like what? Especially, guys, we make use of restriction enzymes. Oh my god, what is this restriction enzymes? And these are going to require restriction sites. Okay, so now let's try to understand. Once again, guys, I want your entire attention out over here. Okay, please be attentive. Okay, see, see, restriction enzymes, they are going to be some proteins like this. Okay, what did I tell you, guys? I told you that we have this plasmid. Okay, and this plasmid, we said, okay, you're going to open this thing up. Okay, and then in that you are going to add your own DNA, right? So you will add your own DNA out over here. This is what we had said. अरे but ये होगा कैसे? There has to be someone who will do this thing for you, right? Who's going to do this cutting and stitching job, right? That job is done by this restriction enzymes. Okay, so cutting job is done by restriction enzyme. Joining job is done by some other enzymes. Okay, that we will see a little later. Okay, now what happens? See, Abhi, this restriction enzyme, guys, please pay attention. Okay, can this restriction enzyme come anywhere and cut any part? Yeah, khacha kach, either way, ada teda. No, right? It cannot do that because then it will not work only. So these restriction enzymes, they will cut only at some specific sites. Okay, these sites we call them as restriction site. Okay, so this is a restriction site. This is a restriction site. This restriction enzyme, it will recognize this and it is going to cut it out over here. It will recognize this part. It will cut it out over here. Okay, so restriction enzymes recognize the restriction sites and cut at that part. In the next case, what will happen? See, in the next case, you will see that that part, whatever that is there, that is going to be removed. Ye gaya bahar. Okay, now there can be many different ways. Like, you can just open it up at one place, but generally we require two places. Ye part gaya bahar, and now this was the DNA of our interest. Okay, so you will have this as DNA of interest. Okay, or we call it as gene of interest. You take this thing and then this is going to be put out over here. Okay, now this, after you put it out, guys, you need to stick this thing also, right? You need to stitch it. So that job is done by DNA ligase. Okay, but then that we'll talk about it a little later. If you have done molecular bases, guys, are you done with molecular bases completely? How is it? How is the situation? Molecular basis of inheritance. Okay, have you talked about DNA replication, protein synthesis? Okay, so you must have come across, DNA ligase, DNA helicase, right? So the same enzyme, basically. Here we're going to use DNA ligase, okay, to join this thing. Anyways, and then you are going to put this thing. So this becomes your, and the gene of the DNA of your interest, or we call this thing as once again your rDNA. Okay, this becomes our rDNA. Okay, everybody got this thing, guys? Okay, important thing that we need to know is we are going to use some special enzymes which are called as restriction enzymes. Their job is to cut the DNA. Their job is to cut the DNA at specific sites. What are these sites called? These sites are called as the restriction sites. Once the restriction enzymes have recognized the site, then they are going to cut down at those places, remove the gene of interest, remove the gene that was or the part of the DNA that was there, and replace it with your DNA of interest. Then the next enzyme is going to come and it is going to stitch it off that way. Okay, so this is what we need to know with regards to your the enzyme part, guys. Okay, everybody okay with this thing? So I'm like making this like in complete layman's term, okay, because slowly as we move ahead, then it is going to be like then we'll have to be talking in detail about it. Okay, chalo. So now guys, out of this, let's try to first see, okay, I just gave you a little example about how things are going to work. Okay, first thing, now let's move into your instruments. Let's try to see what are the instruments or the tools that are used. Okay, so first thing, the tools that are used in your biotechnology, and in that first thing, we are going to talk about some instruments. Okay, so what are the instruments that we are going to use? Now, guys, always remember what is the plan of action. See, always remember your DNA and RNA, they are going to be charged. Now, what kind of a charge is it going to carry? Is it going to be positively charged or is it going to be negatively charged, guys? What do you think? Quickly. What are your nucleic acids? Positively charged or negatively charged? Someone is saying yes. Arey, yes, nahi. Positively charged or negatively charged? Yes, always remember your nucleic acids are negatively charged. And can anybody tell me why are they going to be negatively charged? Because of what? Because of the phosphate group. Hydrogen nahi baba. Hydrogen kaisa negative charge dega? It is because of the phosphate group. Okay, remember phosphate is going to make the DNA negative and phosphate is also going to make the DNA acidic. Okay, so keep this thing in mind, guys. Okay, so now let's give you a little idea. Okay, how do we do? So guys, see, we need to first separate the DNA. We need to separate your RNAs. Okay, and all that thing has to be separated. So in first case, guys, we are going to have our DNA. We are going to have our RNA. Okay, so the idea is, guys, because these molecules are charged. Okay, and especially what charge? These are going to be negatively charged. Guys, we are going to employ techniques which is going to help in the separation of DNA. And what is that technique, guys? So for this, we can use Agarose Gel Electrophoresis. Okay, we are just seeing what are the different techniques. Okay, so Agarose Gel Phoresis. Just a moment. Okay, so now basically in Agarose Gel Electrophoresis, guys, what are we going to do? Okay, now see, our idea is we are going to use several kind of things. Okay, it is not only like on the basis of the charge. We can use like different properties like what is their molecular weight, what is their solubility, what is their absorbance. Okay, so different things can be used. Okay, I'm just showing you one out over here. Okay, now in Agarose Gel Electrophoresis, I'll just give you an idea. Now let's say when you have fragments of DNA. Okay, and I need to separate those fragments of DNA. Now, how will I separate those fragments? So, guys, we have this agarose gel. Okay, in this agarose gel, we are going to keep our DNA. Okay, so let's say these are the wells for putting the DNA. Now, for the time being, just understand as we move ahead, we'll do this thing in detail. Let's say we have a mixture of your DNA. Okay, now how have we got this mixture, guys? See, we said we have this restriction enzymes. So let's say your DNA was something like this. Okay, your entire genetic material like this. We use the restriction enzymes, and restriction enzymes cuts this DNA into your several parts. What have we got? Now we have got, guys, from here you have got एक इतना रहेगा, एक इतना होएगा, एक उससे बड़ा होएगा. Okay, you are going to get different fragments, right? But now in a test tube, guys, will you be really able to see these fragments? No, in your test tube, what will appear is you are going to see all of them as a liquid solution out over here. You won't be able to see array fragments. You don't have microscopy, guys. Now, how do I get the separation of DNA? So what you do is you take this thing, you put it into your DNA. I mean your agarose gel electrophoresis. And now what you do is, guys, you are going to apply charges. Okay. So see, first here you are going to have negative charge, and here you are going to have positive charge. Now, where will DNA move from where to where? DNA will move from negative charge to positive charge, right? And now, why is it? Because the DNA is negatively charged and it is going to be attracted to positive charge. And now, guys, what happens when this is going to separate? See, this has some porous nature. Agarose has porous nature. So what happens is, depending upon the pore size, slowly, you are going to get some bands out over here. Okay. Now, what do I mean by these bands, guys? So what happens is, see, this molecular weight, who is going to be high. Okay. So towards this end, we have high molecular weight. Why high molecular weight? Because see, when the fragment is large, can it travel really towards the other end? No, it is going to be a little difficult. Okay, because it is huge, it has to go through all the pores, it becomes difficult. The smaller one is going to travel till here. The one which is smaller will travel here. The one which is even smaller will go over here. And the one which is smallest will reach out over here. Okay, so this is how generally your agarose gel electrophoresis works. Okay, guys, this is one technique. Okay, and like I said, okay, this is, we are just showing it like, here that it is moving from negative charge to positive charge, and based on their molecular weight and charges, they are going to be separated out. Okay, so how can we, you know isolate the DNA and the RNA by different techniques? Like, for example, like I said, here we are separating them. Okay, these can be separated on the basis of charge. Okay, on the basis of different charges. Okay, on the basis of their different molecular weights. Okay, along with that, like I said, it is going to be also on the basis of their solubility. And finally, guys, we can also, you know make use of their absorbance. That is how much light they are going to absorb. Okay, with the help of that, we can definitely separate these DNA molecules. Okay. Now, guys, here I just said, we have agarose gel electrophoresis. Now, there are two things, okay, in your gel electrophoresis, especially, okay, especially gel electrophoresis. Now, first of all, why do we call it as a gel? Basically, because see, here you have a slab of gel. So this is your agarose gel. That is why we call it a gel electrophoresis. Now, first of all, guys, what do I mean by this electrophoresis? Electrophoresis means the movement of charged molecules under the influence of electric field. That is electrophoresis. Okay, but this electrophoresis, guys, you are going to have two types. We have age, that is agarose gel electrophoresis, and second one we have page. Okay, page is polyacrylamide gel electrophoresis. Age, this is generally done for your DNA. Page, this is, like I said, what first polyacrylamide gel electrophoresis. Okay, this is the one which we generally do it for your proteins. page. Okay, we also call this thing as SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis. Okay, this is also known as SDS page. Okay, now this is one type, okay, which we are talking about, but I'll talk about this thing. So guys, I said, see, they can be separated on the different charges, different molecular weights. We can establish this out over here. But can we separate them by other methods? Yes, this can be separated, okay. By using these properties, we can use another techniques like what? So we can have other techniques like chromatography. Okay, so we have gel chromatography, permeation chromatography, ionic exchange chromatography. Okay, so affinity chromatography. Okay, so we have different types of chromatographic techniques. Okay, these are certain techniques. Okay, so chromatography, like I said, we have gel permeation, we have mass spectrometry. Guys, this is the biggest ever and the most sensitive ever. Okay, mass spectrometry. So, guys, you must have heard that, during this Olympics and all, people do this dope testing, right, to find out whether you have taken drugs or no, or even at the airports, they will find out whether you have taken drugs or no. Guys, the final confirmation is done by this MS, mass spectrometry. This is going to make sure that, even smallest particles can be easily identified that way. Okay. So, So this way. So guys, so first thing is we are going to have this gel electrophoresis technique. And the next thing, guys, the most important technique ever is your PCR. Okay, and now PCR, we need to see it a little bit properly. Okay, but guys, what we'll do is we'll break over here for 5 minutes. Okay, before we move on to the next part, so you just freshen up a bit. Okay, this is a lot of information. I know this thing. Okay, but just just go through this once. Okay, and just see if you are okay with this. If you have any doubts from this part, and then we'll move ahead once again in 5 minutes. Okay, guys, chalo. So let's try to move ahead. Okay, is everybody back? Okay. Okay, chalo. So now, guys, let's try to move ahead and let's try to see what exactly is going to happen next. Uh-oh. Okay, so now I said, guys, like the next important thing, guys, that is going to be your PCR technique. Like this is one of the most important techniques ever. Okay, so what is exactly PCR and what really happens? Who were the people involved in this thing, and how does it really bring about this technique? Okay, so we have this PCR, guys. Once again, like I said, this is nothing but our polymerase chain reaction. Okay. And now, who was the first person to ever talk about this and to give this thing and to prove that this thing works? His name was Carey Mullis. Okay. And later, basically, this guy, he received a Nobel Prize also. Okay. This was in the year 1985. Okay. And this guy, like I said, guys, this guy, he had patented this thing and later then he had to revoke it. Okay. Meaning he had to like give it away for free. Okay, but now, sir, chromatography were uses of no, 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 no. These are another techniques. These are different techniques. So chromatographic techniques are different. Gel permeation chromatography is different. Mass spectrometry is different. Okay, so now, guys, basically, what is the idea behind this thing? Like you take any small amount of DNA, even one fragment of DNA. Okay, and now you are going to run it through this PCR. Okay, so you push it through this PCR, entire system, and immediately you are going to receive billions of copies of DNA within a few hours. Okay, especially half an hour or so, like within a few hours, you are going to get billions of copies. Now we have to understand how are we going to get this thing. Okay, so now, guys, the first thing is we need to see what are the requirements for your PCR technique. Okay, so then we can like slowly move on. Okay, so we have requirements. Okay, so the first thing, guys, the first and the foremost thing that we require is we require this PCR machine. Okay, now what is this PCR machine called? This is called as Pcr Thermal Cycler. Okay, now this thermal cycler. Why? Because it is going to bring about some thermal cycles, meaning temperature changes cycle slowly. So let me just show you how does this machine look like, and then let's try to move on from there. So guys, see, this is how that machine looks like. Okay, so basically this is like the lid out over here, guys. This is a heating lid. Okay, here this black thing, here we are going to put our small tubes. Okay, those tubes are called as Eppendorfs. Again, there is a heating block beneath this which heats, cools that way. Okay, so this is the lid, and here inside this, as I said, guys, we are going to have some heating coils, because rapidly you want to see temperature will increase, temperature will decrease. Okay, all that thing has to happen. Plus there are going to be some spaces. Okay, now I'll just show you this. Here there are going to be these khadda. Okay, these khaddas basically they are to keep your vials. Okay, just a moment. Someone is at the door. Just a moment. Okay, so guys, basically, so this is the orange thing that I'm showing, guys. That is nothing but place to keep your vials. Okay, okay, vial is nothing but a small tube. I will show you what it is. Okay, so this is the place to keep vials. Okay, and these vials, guys, we call them by some special name. We call them as eppendorfs. Okay, now what are these eppendorfs? Basically, see, these are going to be very very small tubes. Okay, so you are going to see that, these are like small tubes like this, and these, they are going to have a cap. Okay, so that, while you are doing or mixing stuff and all, things should not like flow out. So it is going to look something like it's small plastic thing, and inside this now you're going to put your PCR master mix. Meaning what it is, like, the solution, the DNA, the enzymes, the DNTPs. Okay, the forward primer, the reverse primer. Okay, all of them, they are going to be kept out over here. So we call this thing as PCR master mix. Okay, fine. Okay, chalo. So first thing, we are clear, guys. We require this machine. Okay, without this machine, we cannot work this thing out at all. Okay. Now, second thing, what are we going to require? Now, let's try to start thinking slowly, slowly, slowly from our point of view. Okay, guys. So we need to, have multiple copies of your DNA fragment. So first of all, we need to have this DNA fragment, without which we cannot work. Okay. Now, this is the one we need to amplify. Okay. Third thing, okay, after this, see, when we are going to form your new DNA, see, this is your original DNA. It is going to have DNTPs, meaning basically your nucleotides are going to be present out over here. I am going to copy the same thing, and I am going to make copies of this thing. Okay, these are going to be the copies. So guys, for this I will require your adenine, thymine, guanine, cytosine, right? Because the sequence has to be maintained. So see here, if it is, let's say, ATGC, ATGC, I need to maintain that sequence out over here. ATGC, ATGC, right? ये ATGC, ATGC आएगा किधर से? Right, it is not going to be present just like that into your, mixture, whatever that is there. So these I need to add. And what are these, guys? These are nothing but your DNTPs. Okay, so this I need to add. We call them as DNTPs. Now DNTPs means what? Deoxyribonucleotide phosphates. Okay, and nucleoside phosphates, or you can call them as deoxyribonucleotides. Okay, deoxyribonucleoside phosphates. Okay, and phosphate. But then how are these, guys? These are going to be triphosphates. Okay, so the T basically is going to stand for triphosphate. Deoxyribonucleoside triphosphates. Okay, so D N T Ps. Okay, but now who are these DNTPs? This ATGC, it is that only. So it is going to be DATP, deoxyadenosine triphosphate, DGTP, DTTP, DCTP. Okay, so adenosine triphosphate, guanosine triphosphate, thymine triphosphate, and cytosine triphosphate. Okay, so this is how it is going to be. Okay, this is our second requirement, because we need to make this entire DNA, new DNAs. Okay, fine. Next thing. ओके, आफ्टर दिस गाइस, सी, ओके, चलो ठीक है, ये तो हम लोग को बनाने का है, बट ये बनेगा कैसे? लेकिन सी, वी रिक्वायर सम एंजाइम टू create this strand. So basically, when we talked about your DNA replication, guys, which was the enzyme that we used for replication of DNA? It was DNA polymerase, right? So the same thing, guys, here. Now, to create these strands, extra strands, we require this enzyme DNA polymerase. But now, guys, this is not like any normal DNA polymerase. This DNA polymerase, it needs to be heat stable. Why? Because, guys, in this reaction, I'm going to raise the temperature to 91 degrees Celsius. So at that temperature, normal DNA polymerase won't work. Your polymerase won't work. My polymerase won't work. So I need to have polymerase from special organism. Okay, who is going to be heat stable? So who is this organism that is heat stable? His name is TAC polymerase. Okay, so the polymerase that we use, TAC polymerase. TAC, what does this stands for? This stands for Thermus aquaticus. Okay, so this is from the bacteria Thermus this thing. And where does this organism live, Thermus aquaticus? It lives in hot water springs. Okay, it stays in hot water springs. Basically, guys, when you talk about the sulfur springs, the boiling water springs, okay, so we got this organism from there. This generally tends to live at 100 degrees Celsius or more. So if it is stable at 100 degrees Celsius or more, its enzymes are also going to be stable. That is why we took its polymerase, and that is why we use this thing in our experiment. Okay, but now, guys, the next thing that we are going to use, we are going to make use of some primers. Okay, so primers means what? Okay, so let's try to see. So we are going to use two types of primers. One, forward primer, and another one, we call this thing as reverse primer. Okay, forward primer and reverse primer. Okay, fine. So guys, let's try to see this thing. Okay, so let's say we have our DNA. Okay, like this. Okay, now what happens? Let's say this is 3 dash to 5 dash. This is 3 dash to 5 dash. Okay, so something like this: 5 dash, 3 dash, 3 dash, 5 dash, something of this sort. Okay, now, guys, whenever your DNA polymerase works, I want you to tell me, guys, DNA polymerase will work in what direction, guys? In which direction does it work? I want this answer. Without this answer, I'm not going to move ahead. Okay, someone is saying 3 dash to 5 dash. Some people are saying 5 dash to 3 dash. Ah, da, 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 da. Okay. Guys, always remember, you are going to remember, okay, you will remember DNA polymerase as DNA, whatever polymerase you have, we will remember this as DNA polymerase 53. Just remember this as 53. DNA polymerase 53, meaning it will work on, I mean, it will work only in 5 dash to 3 dash direction. Okay, it will not work in 3 dash to 5 dash. Just remember DNA polymerase 53. So now, guys, eventually in this thing, what we will do is we take these two strands, we separate them. Okay, so ye ho gaya separate. Now DNA polymerase will work only 5 dash to 3 dash direction, right? So 5 dash to 3 dash direction, meaning on the new strand. It is not on the old strand. Guys, this is not going to work out over here like this. It is not on the old strand. It is 5 dash to 3 dash on the new strand. Okay, keep this thing in mind. Very, very important. चलो, so now, guys, अभी ये इसको इधर, okay, see, because इसका opposite होएगा ये 5 dash to 3 dash, right? This has to work in this direction, 5 dash to 3 dash direction. अरे but ये चालू कैसे होएगा? Guys, getting this thing, there is a problem. There's a problem. How will it start? How will this DNA polymerase recognize कि मुझे इधर से start करना है? So I need some clues for that. I need a clue, and that clue is given by this forward and reverse primer. Okay, so what will happen, guys? See, one primer. Okay, what is a primer? Once again, guys, it is a small sequence of nucleotides which is complementary to your DNA, the strand of DNA. Okay, so this is our forward primer. Because why? Because this is going to go into the like 5 dash to 3 dash direction. what will happen is, guys, it comes and it sticks out over here at the 3 dash end of the old strand. मतलब ये बन गया 5 dash end of the new strand. This DNA polymerase comes and it sees, अरे ये 5 dash end तो open है, इधर तो है कुछ, तो now it will think that, oh चलो चलो चलो, let's make a new strand. So this DNA polymerase, now it comes and it starts making a complementary strand on this way. So this goes 5 dash to 3 dash direction. Okay with this thing, guys? Fine. So this becomes our forward primer. Okay, so I'll just mention this. Okay, so this becomes our forward primer. Okay, but now, guys, the same thing, I need to go from 3 dash to 5 dash. ये वाला भी हम लोग को ये करना है, right? We need to make that thing also, right? Now what happens, the reverse primer, you are going to see the reverse primer is going to come and it is going to fit itself out over here. So now when it fits itself out over here, what is this end going to be now, guys? This end is going to be 5 dash end of the new strand, right? Now what happens, once again, DNA polymerase, this is going to come and now this is going to recognize that, oh, this 5 dash end, this is open. So chalo, let's make a new strand. So it starts making a strand in this direction. So 5 dash to 3 dash. Everybody getting the hang of this thing, guys? Why we have used this reverse and forward primers? So as to act as clues that from this point you have to start making your DNA. Okay, has everybody understood this thing, guys? Just quickly reply yes or no, yes or no, yes or no, guys. Okay, now what has happened? Okay, now apart from this, guys, what else are you going to require? You are going to require magnesium ions. Okay, so why magnesium ion? Because they act as a catalyst, okay, to bring about this entire reaction. These are your requirements. Okay, so everybody okay with this thing? Fine. So these are our requirements. But now let's try to see how is this process actually done? Like what exactly is going to happen? See, I told you in this PCR machine, we are going to have that block and that block is going to heat, it is going to cool, it is going to heat, it is going to cool. We are going to have cycles of that. Okay, so let's try to see what will happen. Okay, so guys, when I talk about one PCR cycle, okay, so one, okay. So guys, when I talk about one PCR cycle, let's try to see what is it really made up of. Okay, one PCR cycle, guys, it contains of three important steps. First one, we have denaturation. Okay, second one, we have annealing. Denaturation, annealing, and the third one, we have polymerization, or we also call this thing as your extension. Okay, so denaturation, annealing, polymerization. Okay, and now guys, this is one cycle. This is going to last for around 3 minutes or so. Okay, and like this we are going to like 3 minutes, like, or sometimes it is going to be like 1.5 minutes to 2 minutes also. Okay, so it depends. And like this, guys, we are going to bring about around 30 cycles, 30 to 35 cycles. Okay, okay, for a proper this thing, generally the maximum we say is like 30 to 35 cycles, because after that the entire amount of DNA becomes so huge that then the reaction, the sensitivity of the reaction, it decreases. Okay, now once these 30 cycles have been done, okay, now what, I will add one more final step. Okay, so after, okay, after 30 cycles, like 30-35 cycles, whatever you will say. After 35 cycles, you will add last step, that is final extension. This is going to last for roughly around 4 to 5 minutes. Final extension. Okay, so this is your entire PCR cycle. Okay, so let's try to see once again what has happened out over here. See, we have your PCR cycle. We said one PCR cycle consists of three steps. We have denaturation, annealing, and polymerization. So we will repeat this thing like around 30 times. Okay, this will take around 40 minutes or so. And then after that, we are going to have one final step for all. Okay, after 30 cycles, one final step that is called as final extension. That is going to last for 4 to 5 minutes. Okay, now let's try to see what exactly is going to happen. Okay, now generally the temperatures that we will use, it keeps on changing. Okay, according to your protocols and all, it keeps on changing, but I will give you some general idea. Okay, so just to give you a brief idea, guys, denaturation, what does this mean? Denaturation meaning breakdown of your DNA at high temperature. Breakdown of double stranded DNA, I'll just make it more specific. Breakdown of double stranded dsDNA to single stranded DNA. Okay, DS is for double stranded, SS is for single stranded DNA. Annealing step, guys, this is attachment of your primers to DNA. Attachment of primers to DNA, then both your forward primer and the reverse primer. And polymerization, this is going to be formation of new strand. And how are we going to make use of this? I mean, how are we going to form this time? With the help of Taq polymerase. Okay, so that way. So now, guys, let me just show you how thing is going to be, and we will see it. Like, I'll explain you how this works with us. Oh, sorry. 1 minute. Let me just now put my picture down out over here. अरे ये क्या हो रहा है? हाँ. Okay, so guys, this is actually the entire process of your PCR. Okay, so just to make you understand. Okay, first thing, guys, like I told you, we have our DNA sample. Okay, that we are going to take. Then we add our primers, forward primer, reverse primer. We have our nucleotides. We take our Taq polymerase. We take a mixed buffer along with your Mg2 plus inside of it. And then we take this and put it into this tube. Okay, this tube is what? The Eppendorf. We close the lid and now we put it into this machine. Then we are going to see this thing happening: denaturation, annealing, and extension. But now what exactly is happening? Let me just explain. Okay, and guys, once again, please pay attention. Okay, otherwise you will just skip this part. You will not get it. Okay, so guys, when we start off with our DNA, okay, how is our DNA fragment, guys? Our DNA fragment, it is double stranded. Okay, so you will see that, when we start off with our DNA will look like this. Okay, something like this, double stranded DNA. Now what happens? First thing, we are going to enter into denaturation. I told you what is denaturation. It is breakdown of your DNA from double stranded to single stranded. Now generally, what is the temperature that we are going to bring this about at? This is going to be roughly about 90 to 98 degrees Celsius. Guys, really, really high. Okay. But now, At such high temperature, what is going to happen, guys? You are going to see that the two strands of DNA, they are going to be separated. How? The hydrogen bonds in between them, they are going to break apart like this. And now you are going to see that from double stranded DNA, we are going to get single stranded DNA. So I'll just mention, this is going to result into breakdown of hydrogen bonds. Okay. Now, in the next case, guys, what is going to happen? See, we are going to move into your second step. The second step is nothing but your annealing. Okay. Now, annealing generally brought about at 55 degrees Celsius. But now, okay, for you guys, okay, this is nothing but joining of the primers. This is going to be like a range 40 degrees Celsius to 60 degrees Celsius. Now, always, we need to find out what is this range. Okay. Keep this in mind. Mostly we carry this thing out at 55 degrees Celsius, because at that, things happen like really smoothly. So guys, once again, what was the situation? See, we had your double stranded DNA. Your double stranded DNA was half open like this. Now it is converted into single stranded DNA. Okay, so it is open. Now in this case, we will see that our primers, they are going to come. Okay, so we had our primers. नहीं ये नहीं कलर यूज करते, ये कलर नहीं, ये कौन कलर है? ठीक है चलो, I'll use this. Okay, so now what will happen, guys? See, we had our primers. Okay, so let's assume these are our primers: forward primer, reverse primers. Okay, out over here. During your annealing, guys, see, I told you what are these primers. They are going to be complementary to like some sequences of your DNA that you want to amplify. So these primers, they will come and they will attach themselves. So now you will see that, oh, one primer, it comes out over here and it attaches like this. And wherever it attaches, guys, it is going to form hydrogen bonds with this DNA. So then only it is going to fit properly. Same way, the other one, it is going to come out over here and it is going to fit out over here like this. This step is our annealing, guys. Okay, so now our hydrogen bonding is done. So now the primers have been placed. Now, guys, what happens? We are going to go into our third step. Third step is what? It is your extension. Okay, now extension, I told you this is nothing but polymerization. So in this thing, our DNA polymerase, this is going to come and it is going to play its part. Okay, but first, what temperature are we going to carry this thing out? Generally, we carry this thing at 72 degrees Celsius. Okay, for you guys, we are going to use a range 70 to 75 degrees Celsius. Generally, 72 degrees Celsius. Now, in this case, what is going to happen? Let's just figure this thing out. Okay, so now what happens, guys? We are going to have a Taq polymerase. So the Taq polymerase, let's imagine it is out over here. Okay, and this Taq polymerase, it is still going to be active. Okay, really, really active until 100 degrees Celsius. Okay. Okay, so this Taq polymerase, guys, now this is going to come and it is going to recognize the primers. The moment it recognizes the primer, guys, what is it going to do? Immediately, it takes all your DNTPs and it starts putting them across in this direction, 5 dash to 3 dash direction, guys, like this. So you're going to put it this way. Similarly, guys, there is another molecule of your Taq polymerase also, right? So that is going to come, it is going to recognize this. And as it recognizes this, once again, it is going to form new strand formation out over here. And while this is happening, now see the hydrogen bonds and all, everything is going to be placed once again back in position. This is how it is going to happen. Okay, and now, guys, see, if you are seeing In one cycle, what have we got? From one DNA, we have got two DNA. Now, how will this happen, guys? See, two DNAs in the next case, four. Four will give you eight. Eight, 16. 16, 32. 32. Now, initially, you will think, sir, but ये तो बहुत slow है. No, 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 no. Let's see as we move ahead. 32, 64, 64, 128, 128, 256, 256, 512, 512, 1024. Guys, now you will see, yes, absolutely right, Devang. It is now going to be 2 raised to 30. That is humongous. Guys, until here it was slow, but after this is going to be exponential. Just see this thing, 2048. ये फिर क्या बनेगा? 4096. 4096 का बनेगा कितना? Just do it, dude. Multiply it by 2, guys. 8192, guys. And just after this, just see, it is going to be exponential growth. You are going to do this until 30 cycles, and within that you're going to get billions of copies of it. Getting this? So guys, this is the power of your PCR, and that is why this is going to be so sensitive, such a wonderful technique. And guys, now we will do this thing for around 30 cycles. We are going to get billions of copies of DNA. But now, guys, see, I told you, see, after having these three cycles, lastly, what am I going to do? I am going to have this last cycle, that is your final extension, right? So now, what is this final extension? See, guys, after doing this extension, again, this is going to last for like, hardly 30 minutes. 30 minutes gap. Now, a final extension step will last for 30 seconds or 40 seconds in one cycle. In that, guys, are are we really going to be sure that all the DNAs have been completely formed? Is this tag polymerase really going to work at such a great speed? We don't know. Maybe, maybe not. So what we do is we will give the last step, that is the final extension, just a little time for the tag polymerase to complete whatever strands that were left incomplete. So in the next case, guys, you are going to say final extension. This is going to be once again carried out, okay, at 72 degrees Celsius for 4 to 5 minutes. Okay, and now, guys, what happens? Okay, this is max, huh? 4 to 5 minutes max. इससे ज़्यादा नहीं, otherwise, it's going to land up into problem. So in this case, guys, some DNA strands, they were left a little incomplete. Okay, so let me just show you. ये थोड़ा सा incomplete रह गया था. Okay, so ये सब बन गया है. But then some of me may have left incomplete. So I need to form that thing properly. Okay, ऊपर का नहीं, but okay, I need to form this thing. I need to finish that expansion process, and that is why we are going to have this. So why do we do this thing? To complete polymerization of all strands. This way, guys. After this, what are we going to get? So see, where was this happening? This was all happening in the Eppendorf. So after this had happened, guys, we have got our DNA, and this DNA is completely amplified. And now, what are we going to do with this DNA? Now, see, this DNA, it will appear in your Eppendorf like this. Okay, so after some time, you will see that, oh, that Eppendorf, okay, either ho gaya hai, either, that reaction has happened, and the number of your DNA molecules have increased rapidly. Okay, so this is your DNA. Okay, the amplified DNA. But how do we know that the amplification has taken place properly? whether the same DNA has been amplified or some else has been amplified. So guys, what we do is we take this amplified DNA and we run age, Agarose Gel Electrophoresis. So when I run age, what will happen? Let me just tell you. Okay, so the same thing electrophoresis that I had shown you. Okay, so we have these wells once again. Now what we do is, guys, so what we will do, okay, in one well I will put unamplified DNA. Okay, so the original DNA, unamplified DNA, and in the other one here I am going to put amplified DNA. Okay, now what am I going to do, guys? I am going to run this thing. Okay, there is the buffer, then you have electric, the power pack that we call it as. Okay, so now what happens in the influence of electric field, guys? Now this DNA starts getting separated from high molecular weight to low molecular weight. Okay, this is high molecular weight, here it is going to be low molecular weight, and plus how is it is going to be your negative charge to your positive charge. Okay, after some time, let's say you will see that, oh, here there is one band of your unamplified DNA. Now what happens if the same band has been, meaning the same part of the DNA has been amplified, guys, what you will see in this amplified DNA, you will see here there is bhadak band, ekdam bhadak. Okay, dark band. If it is dark band, guys, what does this mean? Here the quantity is more. See, the position remains the same, but here the quantity is more. Okay, so I can definitely say that yes, only the gene of our interest or the part of the DNA which we wanted, only that has been amplified. Okay, now after this, we have to put this thing into your organism E. coli also. So now what are we going to do? This is now going to be removed from the gel. Okay, this is removed from the gel. After that, you remove the gene from it. Okay, how we will see it by using restriction inside, and then you insert it into your cloning vector, that is your plasmid. And once it is there into your cloning vector, now you just take this thing and you put it into your desired cell, whatever desired cell, E. coli, this thing, whatever that is there. Okay, so this it will go into the, see, plasmid. Why am I saying plasmid? Because mostly we will use plasmid only. Okay, that is why. Cloning vector plasmid, take this plasmid, put inside a competent cell, and that's it. Voila, you have got your recombinant cell. And then the cell produces the product. Okay, so guys, this is how your entire PCR is going to run. Okay, I just hope everybody has understood what has happened, guys. This is something different, but only if you understand, then only you will remember. Otherwise, this is all bonkers. Okay, so guys, has everybody understood what has happened out over here? And guys, once again, what I want is I want you to go through this thing, whatever we have done today itself. Okay, because by Thursday, this is all going to just poof, poof, poof. It's going to vanish it. Okay, and make sure you read through the textbook also, because in textbook it has been given in the worst possible manner. Okay, in absolutely worst possible manner. Okay, chalo. So guys, once again, what we'll do, we'll stop over here for a day, and I'll see you in the next class. Till then, take care. Bye bye, everybody. Bye bye.